The Purdue University College of Agriculture has a long history of impact in research, education, and cooperative extension to improve agriculture and the role of agriculture in our society. One of the ways that we continue to do that is in this realm of digital agriculture. It's the fusing of our physical and social worlds to make improved data-driven decisions and actions. So you'll see a website up there in the corner. We encourage you to visit that, purdue.ag/digag. I'm Dennis Buckmaster, a professor of agricultural and biological engineering, and I also serve our dean and our associate deans as a Dean's Fellow for Digital Agriculture to coordinate activities in the college, at the university, and through to external stakeholders regarding data-driven, technology-intensive agriculture. One of the projects that we have is the Wabash Heartland Innovation Network. The WHIN project is not exclusively a Purdue project. It is a partnership with WHIN, the organization, the community foundations in a 10-county region of west central Indiana, of course Purdue, as well as Ivy Tech Community College, and many partner organizations, farms, manufacturing firms, etc. The focus of the WHIN project is to increase the role of advanced technology in agriculture and manufacturing. In this series of brief presentations, you're going to learn of how the WHIN funds are being used to advance the technology in agriculture. So whether this particular video covers the capturing, the clustering, the collecting, or the computing, you'll have to wait to see. But in each of these cases, we are moving forward the technology in agriculture through digital means.
Hi, my name is Suraj Mohan, and I am a second-year master's student within the Agricultural and Biological Engineering Department at Purdue University. I work in Dr. Mohit Verma's lab in Burke Nanotechnology Center, and today I'll be talking about my group's work on developing a cheap, user-friendly biosensor for detecting the presence of bovine respiratory disease in animal samples, with a specific focus on beef cattle. Bovine respiratory disease, or BRD for short, is the most common disease to affect beef cattle feedlots in North America. It is essentially an umbrella term that covers the various viral and bacterial infections that can occur within the respiratory tract of cattle. BRD not only causes serious health burdens for livestock, but also introduces financial burdens for farmers during the process of treatment. The current common practice to diagnose BRD involves the use of physical examinations on beef cattle that are suspected to have the disease. First, animals are monitored for behavior that may indicate sickness, such as having their ears tucked down, being frequently off the feed, and isolating themselves from the rest of the herd. If any animals exhibit these traits, they are pulled from the herd and then given a physical examination for more specific symptoms of BRD. If the sick animals have symptoms such as elevated temperatures and heart rate, significant nasal discharge, and coughing, it is assumed that the animal has BRD. However, because these symptoms can also be caused by other ailments or unrelated reasons, this can lead to inconclusive results in the exam. Regardless, for treatment, the farmer or vet would then likely adopt a trial and error approach by giving an antibiotic cocktail to the sick animal, observing the overall health status over time, and either stopping treatment if the symptoms disappear or prescribing a new antibiotic if they persist. Unfortunately, this style of treatment has been shown to fail up to 33% of the time and can result in large expenses if treatment persists long enough. What makes matters worse is that this treatment can result in the emergence of antibiotic-resistant BRD pathogens, which can spread the disease to more cattle and make the problem even harder to solve. Our lab aims to address this issue by creating the system Discover, which would allow a farmer or vet to screen a freshly collected nasopharyngeal sample for multiple BRD-related pathogens, and based on what the agent or organism and whether antibiotic resistance is present, select an appropriate treatment to give to the cattle. Discover, which stands for Diagnostic Sensors for Cause of Bovine Respiratory Disease, is comprised of three components: our detection mechanism, our physical device, and a result reading system. Our mechanism utilizes loop-mediated isothermal amplification, or LAMP for short, which is a technique very similar to PCR that can easily detect the presence of specific pathogenic DNA in a sample through DNA amplification. However, LAMP only needs to be carried out at a single temperature of 65 degrees Celsius, has higher sensitivity and specificity, and can generate orders of magnitude more copies of DNA than PCR, making it more amenable to visual detection. These reactions would be carried out in individual zones on a paper microfluidic device, as seen in the bottommost layer in the middle of the slide. When the user is ready to test a sample, they would simply add a few drops on the top layer and allow the sample to flow through predefined channels marked in white in the middle layer to the multiple reaction zones. The device would then be placed in our reader and heated to initiate the LAMP reaction. If DNA of a BRD-causing pathogen is present in the sample, the LAMP reaction will successfully occur and create a positive signal on a reaction zone, either through a change in color or a change in fluorescence. In our final device, each reaction zone will be designed to test for a specific pathogen, allowing Discover to screen for multiple BRD-causing pathogens from a single sample. Once the reaction has had enough time to occur, roughly between 30 to 60 minutes, our reader would then take a picture of the reaction zones, use an image processing software to measure any changes in colorimetric or fluorescent intensity of every zone, correlate all of these changes to the presence of certain pathogens, and then finally communicate pathogenic presence and treatment options to the user either through a touchscreen on the reader or a smartphone app. Not only would Discover allow a farmer or vet to conduct BRD diagnostic tests on their own schedule, either at home or in the office, but it would only require a one-time reader purchase of around $100 and subsequent device purchases of $5 per device to do so. Now I will be going into detail of what we have been able to accomplish so far in the three components of Discover. For our detection mechanism, we began with creating LAMP primer sets, which are multiple short DNA sequences that are designed to bind to a unique target DNA sequence and allow for the LAMP reaction to amplify that target DNA. We have been able to create, screen, and select unique primer sets that can target 3 different bacteria that are commonly known to cause BRD, as seen on the table. Our approach consists of finding 3 genes that are only unique to a chosen bacteria and creating LAMP primer sets that only target and amplify the specific gene. These sets were then screened against each other by running the reactions on their target organisms, assessing their various reaction parameters, and selecting the sets that perform the best. This multi-gene approach to primer set design essentially gives us redundant checks that let us confidently say whether a pathogen is present while minimizing the chances of false positives or nonspecific amplification from creating inconclusive results on the test. In this case, we have screened 27 sets and selected 9 optimal sets that would be used on the device. Once we chose our primer sets, we then began optimizing their conditions and tested their specificity. By altering our reaction protocol, we were able to run LAMP reactions that gave little to no false positive results in the data. We then tested our primer sets with DNA of other BRD pathogens to ensure that they would only target and amplify pathogen DNA that they were designed to target. As you can see from the amplification plots, we were able to develop LAMP reactions that only amplified their target BRD pathogen with no false positives being generated. Our next step is to test the detection limit of these reactions and see how they compare to the standard BRD laboratory assays like PCR. Moving on to our device fabrication, we have been able to use colorimetric indicator dyes to easily visualize LAMP reactions under the naked eye. In this case, we can generate a strong change in color from purple to blue for a positive reaction in a solution format. In addition, we have also created some mock designs for our physical device and demonstrated that we can achieve uniform fluid flow and distribution from the top sample inlet sites to the bottom reaction sites. Our next step here is to work on getting these colorimetric LAMP reactions working on the paper-based reaction zones and getting the full device test running smoothly. For our reader development, we have been able to create the image processing software capable of recognizing distinct shapes, in this case such as our reaction zones on the device, and extracting the color intensities within these shapes. In addition, we have developed a prototype reader that would be similar to what a farmer or vet would own and use. The device would be placed on the rolling white tray and be positioned above a heating element that would provide the heat to sustain the lamp reactions. The camera, located on the red lid bottom, would take a picture of the zones, and the final results would be displayed on a touchscreen on the front. Future directions here would be to test our software on functioning devices to test its accuracy, and run our fully functional devices on the reader and optimizing the overall operation of the test. And finally, allowing local beef cattle farmers to test our completed prototype system and provide useful feedback to optimize and refine it for easier use. And with that, I would like to acknowledge all of the hard work that our lab has placed on this project, especially the names in bold, and thank you for listening to my talk.
Suraj, thanks so much for that very nice presentation. I have just a couple of follow-up questions to help us understand the kind of work that you're doing. Since you're detecting DNA, is a starting point sort of the genome sequence of those diseases, or where do you start?
So basically where we start is whatever disease that we're trying to detect. In this case, it'd be bovine respiratory disease. We would look at literature and see which bacteria or viruses are known to cause this disease or show up most often in these types of disease cases. Mm-hmm. And then we usually go on a genome database. So in this case, we use NCBI genome and look at all the available genomes for those specific bacteria and viruses. And then essentially what we do is we do comparisons of all these genomes amongst these different bacteria and viruses and try to find very unique sequences in them that were only present in the specific bacteria.
Okay. So it is very much data-driven, for
yes.
Sure. Yeah. And these DNA that you're trying to detect, how specific is the detection? Is it to a very particular strain of a virus or a bacteria, or could it catch a family of them? I don't know if I'm using the right terminology, actually, but...
So, yeah, that's a good question. So I think with the current data that we have now, we may not be able to get strain-specific bacteria just because in a lot of cases of bovine respiratory disease, it can be caused by various different subtypes of the same bacteria. Mm-hmm. And so trying to find very unique sequences for a specific strain, I think when you're making a diagnostic test like that, you can end up taking a lot of screens in order to figure out specific strains. So I think for this specific project, we're just focusing on species level, and if we can get more specific, maybe down to the family.
Okay, good. I'm thinking that sounds like a better situation than being too specific in the first place. Is that right?
Yes, that's definitely right. Yeah. We've actually seen in some literature that there are some bacteria that are actually hybrids of two separate bacteria that we're testing. Mm-hmm. And so if you were to, for example, run a test on that hybrid, you'd expect to see positive results for two different bacterias. And so that kind of lends itself to why we designed this multi-gene detection approach. So in the case we get these hybrids that aren't really well studied, we do have other markers we can double check to make sure that, okay, it's this type of bacteria that is causing the disease in a specific animal.
Okay, very good. And now that you're working through this and we sort of see the end in sight where it's actually going to work on the farm, what other diseases or scenarios might be solvable with a similar approach, whether they be plant, animal, or human? Are there other applications that could use the same approach?
Certainly. I think you could use this approach for a lot of different applications. And I think because we're simply just using a DNA-based detection approach, as long as whatever specimen you're trying to target or source of the disease, if it's DNA-based, it's more or less likely to be applicable for this device. So to maybe give some more specific examples, human healthcare, if you're trying to screen for immunodiseases that are uniquely genetic in how they are passed on and characterized, use the system for that. Any form of bacterial or viral infections or diseases caused by them, you could use this type of system. And if you really want to get maybe outside of the healthcare side of things, for a really good example would be crop protection. So if you have these patented GMO crops that you want to test to make sure that their primary genes are present and are in the appropriate locations where agreements and purchases have been made, you can use this test as a screen to make sure that whatever crops that are present in a certain field are the crops that were designed to be there.
Okay, I can imagine that there are many, many people who would love to see some of this available to them. So maybe a little bit more of a personal angle. Of course, the work that you're involved with is a relatively large team. You showed us that on your last slide with the credits. What's your particular part of this project?
So my particular part, I kind of play two roles for this project. So the first one is actually doing the work of characterizing all of these different primer sets that we're using in our detection mechanism. So seeing what the properties are like, and then based off that, using some basic data analysis through MATLAB, I would pick out which primer sets are the best or which ones would be used in the final device. And then my second role is actually coming up with designs of the physical paper microfluidic device. So figuring out how I want that layout to be, where do I want all these detection reactions to be placed, and then essentially making the prototype devices we would test with our reader or in any other way.
Well, it certainly is exciting work, and I appreciate the update that you've given us.
No problem. I'm glad I could share some of the research that I do.